Okay, so last time we left off on this right here. Wow, that is taking forever. Okay, so some DNA molecules, they can be extremely large, of course. You already probably knew this from previous classes and little fun factoids, right? Like you can stretch out all your DNAs end to end to hit the moon or something. Okay, so humans have approximately 3 billion nucleotides in each strand of our DNA. This is divided among 24 chromosomes, 22 of which are autosomal, 2 of which are sex chromosomes, the X and Y. If you're a male, you have what? X and Y. And if you're a female, you have? Double X. You got 2 X's. Fantastic. Okay. The Indian muntjac actually has a similar sized genome that's distributed on 3 chromosomes. So instead of having 24 chromosomes, it just has really, really long ones that has 3 chromosomes. Okay? Corns have been found with 400 something, 800 something sets of chromosomes, in case you're wondering. Okay. Anybody have any questions about that so far? That's something you've probably heard before. If you have trouble remembering this, there was a famous company called 24andMe. It's called 24 because that's the number of chromosomes. They did genetic testing. And apparently they sold your genetic profile. That's creepy, huh? Note to self, don't do 24andMe. Okay, so DNA chains do have directionality, or another way of saying that is they have polarity. When one end of the free 5' OH group and or a 5' OH group attached to a phosphoryl group, this is one end of that 5' side. The other end is going to have a 3' side. This could be a free OH group if you're at the beginning. By convention, you would write the nucleic acid sequence. They're going to be written from left to right, and so they'll be written from the 5' to the 3' direction. Okay? And so typically when you're looking at a chain of DNA, you would orient it such that what is in the front? Oh, that's a tougher question, isn't it? Okay. Okay, so when you're looking at this, if you were to look at DNA, what you'll see is that the 5' would connect to the 3', the next 5' would connect to the next 3'. Okay? Okay. And so you read it from the 5' to 3' direction, don't you? Okay, fantastic. Okay, so the nucleic acid chains are going to be represented by only the identifying bases. We typically do not look at the back structural part of the chain, okay, which is the sugar and the phosphate or phosphoryl, depending on if bonds or not. And so this is an example of one chain of DNA represented as a one-dimensional structure, right? And so we have just written the bases. This is the adenosine, so on, right? A, C, A, T, 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 T. You got it. Okay. Is this DNA or RNA? What do you look for when you're looking for the difference? Yeah, it's exactly. You look for a T, right? Yeah. No T, a U instead. We have RNA. Okay. Okay, so these strands are going to pair with a complementary sequence. This is going to form a double helical structure. The double helix is going to form because the bases on 2 separate nucleic acid strands are going to form specific base pairs. That's that ATCG that you probably are familiar with, right? And this is going to be stabilized by the hydrogen bonds, but also the van der Waals interactions, which oftentimes get forgotten. Those are those dispersion force interactions, by the way. This is going to facilitate the replication of genetic material. This is why this works as a pattern or blueprint, right? Is because you can copy the exact sequence over and over by matching up the bases. All right, so this is actually an X-ray diffraction photograph of a hydrated DNA fiber. You can actually see some nice patterning on it, can't you? By the way, in case you're wondering, this with Linus Pauling actually almost got the DNA structure correct. He just had the bases pointing in the wrong directions. Yeah. And then the Watson and Crick. So the features that were deduced from that diffraction patterning are two helical anti-parallel polynucleotide strands are coiled around a common axis in a right-handed helix. The sugar phosphate backbones are on the outside, and the purine and pyrimidine bases rely on the inside of the helix. The bases are going to be nearly perpendicular to the helical axis, i.e., the bases are lining up here and twisting around in the middle. Okay? The adjacent bases are actually separated by about 3.4 angstroms. The helical structure is going to repeat every 34 angstroms. This is going to be about 10.4 bases per turn of the helix. There is a rotation of nearly 36 degrees per base. The diameter of the helix is about 20 angstroms. Now, Watson and Crick, did they actually do the data that solved this? No, they actually stole that from their neighbors, right? Which are two female scientists. Yeah. Though they did do other work later that was very Nobel Prize worthy, but yeah. So there's actually a kind of shitty story behind this. I'm not going to go a lot into that. There's plenty of documentaries and stuff that cover it. Okay, so this is the Watson-Crick model of the DNA. Here's the side view of it. So right here we can see this is that helical structure. You see your bases, hydrogen bond right here. This is your axis of your DNA along its length. And what we can see here is the bases actually stack on top of each other, don't they? Yeah. Okay, does anybody have any questions about that? Okay. This would be looking down the DNA, and you can see the sugar phosphate backbones actually have a rotation that happens around like a little corkscrew or something, right? Okay, so again, this is the structure of your base pairs. This is probably the most important part of what Watson and Crick actually did, by the way. This overall structure here was actually proposed by Linus Pauling, with the only difference being the base orientations. That's the only thing he got wrong. And then the diffraction data was taken by the neighbors of Watson and Crick. What the important part of what they did was they matched those up and figured out the bases are pointed like this towards each other. Okay? So when the guanine pairs with the cytosine here, and the adenine pairs with the thymine, which is right down here, the base pairs have essentially the same shape overall. See that? They line up very nicely, right? This is what allows that to have the repeated helical structure where they stack nicely on top of each other. So the base pairs are actually going to be held together by weak hydrogen bonds here. And yes, they can be melted. And that would be what melting DNA is, right? Breaking those hydrogen bonds. Okay, so in the 1940s, Erwin Chargaff actually observed that the AT and GC ratios were nearly 1-to-1 in a variety of organisms, while the AG ratios varied greatly. And right here we can actually see this work. And again, we see the AT ratios are basically always around 1, right? We see the GC ratios, they're almost always around 1, though you can get a little bit of difference down here in that organism right there. What we see is the AG ratios, on the other hand, vary a lot, right? Okay. Why is that? If you saw a pattern like that, what does that indicate to you? AT has to be paired somehow, doesn't it? So it has a paired relationship, right? GC, again, has to have some sort of paired relationship, doesn't it? Except for that organism right there. And AG They have to not have any pairing relationship, do they? Because they're always different. Now, if you actually got something like a 1.5 and all these are 1.5, or a 2 here and all these were 2, that would still mean it's a pairing relationship somehow, right? That they're related. Okay. They didn't have to be 1-to-1, but they are in our case. By the way, an interesting side note is they have made trimer DNA, and apparently that is something that could have been existed. So, just kind of weird, right? Let me just blow your minds here. Okay, so remember that these base pairs are actually going to stack. When these base pairs stack, they're actually going to make van der Waals interactions. And this is actually one of the key things that makes this such a stable structure. If it was just the hydrogen bondings, it would not have the stabilities it does. So this base stacking is again going to contribute to that stability of your double helix. And the reasons for this is that the double helix formation will be facilitated by this hydrophobic effect. That base stacking and slight rotations are going to line these up the best. Okay? The stacked base pairs are going to attract one another through van der Waals forces. Again, that's going to help them hold together in an environment where do we have a lot of? We have water, right? And so water can make what sort of interactions? Hydrogen bonding, right? So if you were to have just these hydrogen bonding happening, would it be very hard for water to split these up? No, there's not that much difference between hydrogen bonding water to those and hydrogen bonding each other, right? Yeah. But what happens is, if you look at these rings, they're not exactly the most hydrophilic substances ever, right? They actually have a fair amount of van der Waals force or dispersion character, don't they? Or another way of saying that is hydrophobic or nonpolar, right? Okay. And so what ends up happening is you get the stacking force, which is what helps you make your helix structure, okay? And then you can't pair a guanine with a thymine because it won't make the proper hydrogen bonding, and therefore it won't have the proper sizing, will it? Okay? And vice versa. And so you get the stacking effects. That makes it make that helical structure where it stacks on top of each other and forms into dimers. And then you get the hydrogen bonding, which is sort of your keying. Okay? And there is some energy that you gain through that, but it's not enough energy that would do it by itself. The DNA, this can assume a variety of different structural forms. We can form the B form, which is the right-handed double helix made of anti-parallel strands held together by the Watson-Crick base pairs. This is the majority of DNA under physiological conditions as your B form. Okay? This is the one you're probably most familiar with. We can have the A form. It's similar to the B form, but it's wider and shorter. It'll also have tilted base pairs relative to your helical axes. So previously, we had our axes, right? Right here. See the bases here? How they're parallel with that, or they're exactly perpendicular with it? See that? Okay. The B form, this is going to be shorter and fatter, but it's also going to have these bases here tilted with respect to that axis. Okay? Can you sort of envision that? Yeah, the A form will have that tilted. The B form is what you normally see because it's the most common physiological one. Okay, this is actually seen in RNA double helices. This is also seen in RNA DNA hybrid helices. So whenever you do that normal transcription process that we've been talking about, this is actually fairly common in that. Okay? Anytime you make a hybrid, it's fairly common to see the A form. The Z form is going to be the left-handed double helix. This will have a zigzag phosphoryl groups. As of the publication of your book, apparently, they don't know what this biological role for this is. I haven't seen anything where they show this in actual use. But the zDNA binding proteins have been isolated. So It obviously has to have some biological function. There's proteins that bind to it. So, something. Maybe you can go off and figure that out, okay? Okay. Has been mentioned, though, because it's definitely a thing. Right. That's the Z DNA. This is actually what these look like. So again, this is the B DNA. This is what you're used to seeing. And everybody can see that from the previous diagram as well, right? Yeah. Okay, this right here is the A DNA. Again, remember, this is the less common form of DNA. This is more common when you see like a hybrid or RNA making a double helix, okay? And again, see how it's shorter and fatter? And we have a tilt. See the tilt to it? Like here would be the axis. And they're all tilted off of it. Everybody said okay? Okay. Okay, this is the Z DNA. They actually did a good job naming this one, because see how it says zigzag? Yeah. And so Z DNA, do you know how to tell the right-handed from left-handed? So point the direction it's going, curve your fingers, and see how it goes in that right-handed direction. See that? Yeah. It's turning in the direction of your fingers, isn't it? Yeah. Okay. If it's left-handed, you would do that with your left hand, and it would do that. And see how it does that? It's curving the other direction. Yeah. Okay. These are right-handed. This one's left-handed. And yeah, don't know the biological reason for that, but apparently it's something. The sugar pucker actually explains many structural differences between the B form and A forms of DNA. This is the sugar pucker, by the way. So in the A form here, you have, right here is your oxygen, this is your base. See that okay? So in the A form, it actually does the envelope structure. Remember the envelope structure from 5-membered rings back in organic chemistry? Hey, I know, confirmations, right? In the B form, it actually does this puckering, which is the opposite. We've puckered this part right here downward into an envelope, okay? It's a little harder to see the envelope in this case, but you can kind of see it right here, right? And then pucker down. So again, in the aDNA, the C3 is going to lie out of the plane. That's formed by the other 4 atoms in the ring. This is the C3 endo form, because the 3 is the one that lies out of the plane. This is going to lead to an 11-degree tilt of your base pairs, which is what causes ultimately that DNA form. So in the bDNA, you'll have the C2, which lies out of the plane. It's a C2 endo form. Okay? Or another way of stating that would have been for it to be the C2 envelope. Okay. This is going to give you your B form, which is that one right there. I'm sorry, did I? Okay, A. Yeah, I went backwards because I was looking at the other. Yeah. Okay, this one gives you your B form. Oh, I'm sorry, wait. Oh, which one? Okay. This one here, C2, gives you your B form, which is right here. This one here gives you your A form, which is this one here. And it's the C3 endo form that gives you your A form, which is the less common one. Your B form, more common one, is this one, which is the 2 endo.
Why am I doing the more common B?
Good question. It's a good question. I don't remember off the top of my head, but I suspect it's the order of proposed model and discovery. That would be my guess. Yeah. That usually is what it is in biochemistry and biology when they do like the factors, first factor isn't first, or A and B form. A form isn't the most common first one. Yeah. I know, right? Would have made my life a lot easier, huh? Yeah. It would have made all of our lives more easy, but biology, people. What can we do, right? I have to be ashamed that actually this is probably a chemistry person that screwed this one up. Because almost all of these people I just mentioned are chemistry people. Linus Pauling was. He got the Nobel Prize for organic chemistry, in fact. So this is the comparison between these, and there are different factors that we just were talking about. Does anybody have any questions on this? No? The major and minor grooves are going to be lined by specific sequence-specific hydrogen binding groups. The major and minor groove will arise from the glycosidic bonds on a base pair that are not diametrically opposed to each other. The major group is actually going to be wider and deeper. That's why it's called a major group. It's larger than the minor group. The hydrogen bond donor and acceptor atoms in the grooves are going to enable interactions with our proteins. This is essential for the replication and transcription. This is actually part of what you probably were missing out when you've seen these processes before. Okay? You may have been wondering, like, why is it doing it right there? It's the major and minor grooves. Okay, so these are the major and minor grooves. So you can see sort of the DNA structure here. See that okay? Okay. And that's that normal, more common, which form? B, you got it. Yeah, see how it's longer and thinner? Yeah, I know. Let's practice a little. Okay, more common B, less common A. All right. You may have noticed before, if you looked up here in the previous one, there's your major groove, there's your minor groove, right? You can pretty clearly see it. Okay. And again, that is right here. Right here, we can see the hydrogen bonds, how they line up with the bases in the middle. One side had less stuff, right? See that? The backbones come out over here. And so one side is larger, isn't it, than the other side. And this larger side always corresponds to your major groove. It gives more access to the bases. Your minor groove side, on the other hand, gives very little access to the bases themselves, doesn't it? It'd be very hard to get at them from the minor groove side. Right here we actually see, again, the GC pairing. And again, we see the major groove on this side, minor groove right here. And that's where we're seeing right here, corresponding. Okay? Everybody see that okay? Fantastic. All right, so some DNA molecules are going to be circular. Some of them will be supercoiled. The DNA molecules, these must be compacted to fit inside the cells. As you saw from that previous figure quite a while ago, where it had the free DNA, and it was much, much larger than the cell would have been, right? Do you remember that? Where it was sort of starting to uncoil? Wasn't even fully uncoiled, right? Okay. So the E. coli has a circular DNA molecule. This is twisted into a superhelix by the process which is called supercoiling. Okay? You've all actually seen this to a degree when you're looking at like wiring, right? Okay, so you have a wire, right? And do you guys use headphones with wires anymore? How about a charging cable? Like a thin charging cable. Okay. Okay. So the thin charging cable has a bit of a twist to it itself, right? And that would be the normal twisting we're associated with the helix. Okay? But it can also then wrap around, can't it? Okay? And that would be like the circular motion that wraps around. But we also get one more thing, which is referred to as supercoiling, right? And that's where you usually end up with your tangles. Where it coils kind of like a hose when you're trying to uncoil it, right? Where it makes bigger coils. that are a further expression of coiling on that structure. Can you all envision your cord tangling up because of the supercoils? Fantastic. Okay, and so we'll make a super helix. So we have the base helix, and then we're going to coil again, where like a pigtail or a phone cord from back in the day, where it makes a super helix. Okay? Can you envision it? Okay, fantastic. And that's your supercoiling. The supercoiling is going to be biologically important for a multitude of reasons, but this is sort of your entry level, which is supercoiled DNA is more compact than relaxed DNA, i.e. we're compacting it so we can store it in a smaller space again, right? Supercoilings actually may hinder or favor the capacity of the double helix to unwind and interact with other molecules. So to transcribe DNA, what do we actually have to do to it? Can we transcribe it when it's all packed away and we can't get to it? No, we have to get at it, don't we? Yeah. And so you actually have to uncoil it to transcribe it, right? To replicate it, what do you think you have to do? You have to uncoil parts, right? Yeah. And so to actually do anything to the DNA, you actually have to partially uncoil it. This coiling process can actually also alter how likely a part is to be read. So you can make the parts that you want to read more often show up more often. Okay, so this one right here would be no supercoiling. See, we have the helix. It's just in a roundish shape, right? This one has supercoiled more. And see the supercoiling? So we have scrunched up and wrapped it even more, right? And then it also has taken on a kind of weird blobbish shape now. So these single-stranded nucleic acids can adopt elaborate structures. We have the stem loop structure, which is a common structural motif that's seen in nucleic acids. This is going to occur when two complementary sequences with single strand form a double helix. This can include mismatched base pairs or unmatched bases, because there's still going to be some energy feedback and energetic favorability to making helix structures, even if we don't have perfect matching, right? I.e., it's better to make a hydrogen bond and make a van der Waals force than it is to not, right? Even if it's not the full three. So more elaborate structures may form. Usually these are stabilized by magnesium 2+ ions. That's a very common component. These actually are the stem loop structures right here. Can everybody see those stem loop structures? So here we have a 5' going in. We have a set of bases that mostly match, okay? And so they have a pretty good stability matching. And it will come up here. It will match until it stops matching really well. It'll then, or we get too far out, in which case our energetics are not favorable anymore. And then what it will do is it will make a loop, because it can't just 90-degree turn, right? We have to make a loop. About 4 base pairs, 5 base pairs is typically the sort of minimum you need to make a loop, okay? And then here we have the RNA version of that. Does anybody have any questions about those? They're pretty much what they say, aren't they? Yeah. Yep, it's a single one of the strands. Yeah, they could be matched up actually into a double strand, though that would make that a lot less energetically favorable. Okay? But it happens a lot more with single strand. RNA, you tend to see this quite a bit because a lot of the RNAs are single strand. Okay, so again, this is again, your complex structure of your RNA molecule here. What's that right there? Yeah, that's our stem loop, isn't it? So the 3 nucleotides that are highlighted, these are actually the part B right here. Okay. And we can actually see these over here making a fairly complex base matching. okay? Which is kind of interesting. Ooh, so beautiful. So our double helix is going to facilitate the accurate transmission of hereditary information. The separation of your double helix is going to yield 2 of the single-stranded templates. Because of your base pairing rules, the sequence of one strand is going to determine the sequence of your partner strand. This is referred to as semi-conservative replication, and the definition of that is a process by which each replicated DNA molecule contains one parent strand and one newly synthesized daughter strand. Why is it referred to as semi-conservative? Yeah, half and half, right? Yeah, semi. Okay. Yep. And so we conserve one half, make one half new. Fantastic. So what does that mean about your DNA, by the way? What does that mean about your DNA? Theoretically, somebody on this planet has DNA from the original. Yeah. Literally would have a strand of DNA from the original, right? Now, it's unlikely it's not changed at all, but somewhere on this planet, we have people that have little bits of strands that are the original DNA from the first human that had just been passed down, right? Yeah. Isn't that wild? Okay, so the differences in DNA density will establish the validity of the semi-conservative replication hypothesis, because of course we're a science class, so what do I have to give you after everything I say? Any hypothesis I say. Proof, right? Yeah, we gotta try to disprove it. So Messelson and Strahl's experiment. Step 1, we label the parent DNA with N15 by growing bacteria in growth media that contains only N15. And so now we have isotopically labeled DNA. Step 2, we shift the bacteria to media grown with N14, which is the normal isotope, right? And so now, in this new media, we would not have any additional, or at least almost any additional N15 brought in, right? So our parent strands are all going to have N15s. Our new strands will have N14s, pretty much, right? At least not statistically they will. So step 3 is going to be perform the density gradient equilibrium sedimentation. This is going to determine the distribution of the N14 and N15. Remember, Ns are incorporated many times. So when you're looking at a DNA with thousands of Ns, then how many, what's the difference in molecular weight? Thousands, right? Yeah, so it can actually add up quite a bit. So the experiment question was, what is the distribution of N14 and N15 in the DNA molecules after successive rounds of replication? And so again, the N15 isotope will be heavier than the N14. These will separate, giving us 2 different bands for the different strands of DNA. The density gradient centrifugation can establish between the DNA that contains N15 and the DNA that contains N14. By the way, would you necessarily have to do a density gradient centrifugation now? We have other techniques we could do now, right? But back in those days, this is a brilliant way of doing it, right? Really smart. Okay, and what we can see here is, we got 2 bands here after this growth cycle, and they're pretty close to the same amounts, aren't they? Yeah. And so 1 generation, your newly synthesized DNA with equal parts N15 and N14. This is actually consistent with your semi-conservative replication. And so first generation 0, we just have N15. That establishes our baseline, right? That we're not having things magically get N14 into this DNA. Okay? So that's the one we grew only with N15 available. The next generation, what do we see? Well, our bands actually go in the middle between N14 and N15. Everybody see it okay? And so what does that tell you? Well, that first generation in the new media, it started getting N14 in here, right? But about even. So we probably have, if we were to make an estimate, 1 strand of each, don't we? Okay? What are we seeing in generation 2, by the way? We get a mixture, don't we? We have some of them which are in 14 only, and then some of them have in 15 strand. Okay? And that's giving you one of the strands from here is matching up to another in 14 strand, right? And then you see that this population starts disappearing each generation, though it never goes away, does it? Okay. Anybody have any questions? Fantastic. This is that semi-conservative replication. This is what we were talking about. Remember, the parent strand is going to be split. We'll have two daughter strands that are attached to those, and that gives us two strands that are between the weights, and if you use N14 and 15, right? Medium weight between. And then what happens next? Well, the first generation daughter molecules pair up with new daughters, and then the parent strand pairs up with a new daughter. And so that gives you 1/4 of the DNA with a slightly enhanced weight, right? Okay, and so on. We can actually reversibly melt the DNA. They are held together by van der Waals forces and by hydrogen bonding. And this means that's a reversible melting. That's literally a physical property change, right? Like melting ice. Okay. So during replication or transcription, your two strands of your double helix must be separated. In the laboratory, the DNA strands can be separated by heating a solution of DNA or adding an acid or an alkali, which can also cause melting, as it were. In cells, the helicase is going to use chemical energy to disrupt the helix. The helicase is going to actually unwind that helix using chemical energy. Melting is going to be the dissociation of a double helix. And you can partially melt the DNA and not fully melt it, in case you're wondering. The melting temperature, which is Tm, is going to be the temperature at which half of the helical structure will be lost. This would normally be half of the molecules losing their helical structure, but theoretically you could have half the helix unwinding, I guess. Annealing, this is the renaturation of your double helix. Notice, by the way, this is literally the same terminology as melting and crystallizing that we would normally use with like a crystal, okay? Annealing is the same terminology exactly, literally making it back into the solid, okay? And this is going to occur when the temperature is lower than the below Tm, which is your melting temperature. Notice that this is literally a melting point temperature for DNA. Same as you probably remember from your Gen Chem 2. Now, the monitoring the melting part, the hyperchromism, this is the effect where the bases stacked in a double helix absorb less ultraviolet light at 260 nanometers than the bases in a single-stranded molecule. The melting can be monitored by measuring the increase in absorption of 260 nanometer light. It's actually very, very obvious. So when you get to your melting point, you get a sort of sigmoidal effect where it goes up really rapidly, okay? And what that means is that at this wavelength, you'll see this really big jump in your absorption, okay? Okay, so right here we actually see the 260, this 220, 300, and here we can see the double helix, which absorbs less, right? The single-stranded, on the other hand, will absorb more, okay? We'll break apart the double helix to two single-stranded, right? And so what's going to happen here is this is, again, your relative absorbance at 260. So we're just monitoring 260 and seeing what the absorbance looks like. All right. So we change the temperature, we increase the temperature, it gets up to about 70, so we get up 65-ish, 64-ish, and what we start seeing is this absorbance starts rising, right? And then it rises quite a bit, 70 would be the halfway point of this, and then we get up here, and it kind of cuts off rising, right? Okay. Or at least it greatly reduces. This would be a logarithmic curve right here. See the sigmoidal pattern? Okay. Okay. So the Tm of this is around 72. degrees Celsius. Note, by the way, 72 degrees Celsius is a temperature we don't normally get to, right? That would be very bad for us. Okay? Why do you think that is? Because our DNA would melt. We don't want melted DNA. That is bad. So your DNA replication by polymerases, that's going to take instructions from your templates. So your DNA polymerases are going to catalyze the phosphodiester bridge that's in the formation in the step-by-step manner. This is the overall reaction that you would see for the DNA synthesis. So we have DNA that is N long, right? So that's the number of DNA bases is N. We then add a deoxy, see the D. What does NTP mean again? It's any of the nucleotides, right? Yep. Yeah, so this would be ATP, GTP, you get the idea. Okay, so we do the reaction. Notice this is an equilibrium because all these processes are always equilibrii, right? Okay, so this equilibrium adds one of the new DNA, could be an ATP, could be a CTP, whatever, right? And this is going to cost it 2 phosphates. See the 2 phosphates coming off here? Fantastic. Notice that this will lengthen the DNA by 1 base pair, right? So +1. So again, don't forget that this is the deoxyribonucleotide. It's the generic form of saying something like ATP, CTP, etc., right? It denotes all of them. The deoxy, if you were doing RNA, this would not have the deoxy part, right? Okay. It would just be ribonucleotide. Again, this is the pyrophosphate ion. This is 2 of the phosphates coming off. This shows you this in a bit more detail. So here we have our primer on here. This is then going to come in, react to an ATP in this case, a DATP. That matches up with the T. And so what we'll do is we'll then form our phosphoryl linkage right here. This will use up the energy from 2 of those phosphates that were on the ATP. Okay? And this sticks properly because it's an A and T matching. Okay. And then here's the GTP doing this, matching up with C. Okay. We keep lengthening it from the 3' chain, don't we? Over and over and over. Okay. Don't forget that during this process, we've also used the helicase on windings. We'll go in more detail on the replication. We'll have a nice little diagram to talk about it. But this is that entry-level information you need to get there. So the key characteristics of your DNA synthesis are going to be things like the reaction is going to require 4 deoxynucleoside, 5 triphosphates. They're also going to require magnesium 2+. What is something I kept saying about anytime you have that phosphate groups and you have to deal with those? What it usually comes with it? Magnesium 2+, right? Yeah. The new DNA strand is assembled on a pre-existing DNA template, the template strand, which is where our parent was accompanying our daughter each time, right? And then we just keep doing a semi-conservative replication. The DNA polymerases will require a primer to begin the synthesis. They cannot actually start from scratch. They need a primer to start off. Many of the DNA polymerases are going to have nuclease activity to remove mismatched nucleotides. And so what typically happens is if we got a mismatch, let's say instead of A, we had gotten G or C, okay? Our polymerase would have actually gotten halted here. It would have been the wrong size. It couldn't move back on. This process is a what? Equilibrium. So if we stay just stuck there with our polymerase, what's going to happen to that? It'll pop off, won't it? Yeah. There you go. The overall, that's how we check the match. If we have the incorrect match, you'll have the wrong size. Okay? And we will have a mismatch nucleotide, and it will remove the mismatch nucleotide. This helps us maintain our order of bases in our new template, our new strand. Okay, so the DNA polymerases are going to catalyze strand elongation. Again, here we see the overall concept. Right, again, we have the primer strand. This is our DNA template strand. Our bases will match each time. Here we're seeing, though, more clearly the mechanism part, right? And so what we have here, this is the hydroxide, or the hydroxyl group on the 3' end, right? Which is the one we're going to elongate. We're then going to have a nice little nucleophilic attack on this phosphate here, which will cause this oxygen to leave as the PPI-free phosphoryl group, right? And then this is going to give us phosphate linkage with the next base, which happened to match this base right here. Okay. Everybody said okay? Fantastic. Note, by the way, that these will leave as PPI, but they'll actually be hydrolyzed later, so you have 2 free phosphate groups. Okay. Anyways, there we go. Fantastic. So the genes of some viruses are actually made of RNA instead of DNA. So some viruses have RNA genomes. These will be replicated by direct RNA polymerases. The tobacco mosaic virus is typically the big example of this, and the one that typically gets used to look at these things. Okay. Retroviruses, these are going to be viruses with single-stranded RNA genomes. They're going to be converted to DNA double helices by reverse transcriptase. And so, for example, HIV-1, the human immunodeficiency virus 1, is a very famous retrovirus, right? And this is how that functions. Okay, so this is actually the RNA to DNA in a retrovirus. So here we have the viral RNA. We have a reverse transcriptase that's going to reverse the transcription process, right? And so we're going to synthesize a DNA complementary strand, which is the blue one right here. This will give us our DNA-RNA hybrid. The reverse transcriptase is then going to digest the RNA. Then we're going to have reverse transcriptase synthesize a second strand of DNA. At this point, the normal replication process would just do this, right? From here on. And will continuously replicate these DNA strands. Okay? Does anybody have any questions about that? Yeah, the overall diagram is pretty much what you expected, right? Okay, so the gene expression is going to be a transformation of your DNA information into functional molecules, which will actually, have structures that will dictate function and do stuff. All right, so the DNA is going to be expressed into 2 steps. We have step 1, which is an RNA copy. This is usually referred to as messenger RNA or mRNA, which is the more common one. This is going to be made that encodes the directions for protein synthesis. You can think of this a bit as, you guys ever worked on a work site or seen people work and build a house or apartment building or whatever? Road. Okay. You have the plans that you submitted to the city office, right? And there are big plans, right? Lots of parts, lots of numbers, very confusing for people that don't know what they're looking at, right? And so what do they do with those? Well, they take parts of those and they condense them down into smaller plans that people working on the site can understand, don't they? And that's what you're doing here. You're condensing them into what you need to actually make what you need, right? Not the whole damn thing, okay? And then I guess we're stopping here.
Yeah.
Yeah. Yeah. that's fine. We can stop here. I think you all know this well enough on this part of it. That's not confusing. Does anybody have any questions or anything? No?